Welcome to our newest episode about the DNA. Subscribe our channel and enjoy. DNA, which stands for deoxyribonucleic acid, is a complex and remarkably long molecule that contains the genetic instructions for the development and function of all known living organisms. This molecule consists of a sequence of nucleotides, which are the building blocks of DNA, arranged in a double helix structure that resembles a twisted ladder. Each nucleotide in DNA is composed of a sugar molecule, a phosphate group, and a nitrogenous base, with 4 different types of nitrogenous bases: adenine, A, thymine, T, guanine, G, and cytosine, C. The specific sequence of these bases determines the genetic code of an individual, dictating traits such as physical characteristics, susceptibility to certain diseases, and even personality traits. DNA is essential for life and has a fundamental role in the transmission of genetic information from one generation to the next. James Watson and Francis Crick are credited with discovering the double helix structure of DNA in 1953, based on data and research findings from several other scientists, including Rosalind Franklin, Maurice Wilkins, and Erwin Chargaff. Their discovery was a significant breakthrough in the field of molecular biology, as it provided a better understanding of how genetic information is stored and transmitted between generations. Watson and Crick's discovery also paved the way for many subsequent advances in the field of genetics, including the Human Genome Project, which mapped the entire human genome in the early 2000s. The shape of DNA is often described as a double helix, which resembles a twisted ladder. The 2 sides of the ladder are made up of alternating sugar and phosphate molecules, while the rungs of the ladder are composed of pairs of nitrogenous bases. The nitrogenous bases pair up in a specific way: adenine, A, pairs with thymine, T, and guanine, G, pairs with cytosine, C, through hydrogen bonds, forming the base pairs that hold the 2 strands of the double helix together. This structure provides stability to the molecule while still allowing it to unwind and replicate during cell division. The double helix structure of DNA is essential for its function as the carrier of genetic information, as it allows for the precise copying and transmission of this information from one generation to the next. The order or sequence of the nitrogenous bases along the DNA molecule determines the genetic code of an organism, which is responsible for its unique characteristics. The specific sequence of bases along the DNA molecule determines the sequence of amino acids in a protein, which in turn determines the structure and function of that protein. Proteins are essential molecules that carry out many important functions in living organisms, including serving as enzymes, hormones, and structural components of cells and tissues. Therefore, the unique sequence of DNA bases in an organism's genome ultimately determines its physical traits, such as eye color, height, and susceptibility to certain diseases. Even small variations or mutations in the DNA sequence can have significant effects on an organism's traits and can contribute to genetic disorders and diseases. The human genome is estimated to contain approximately 3 billion base pairs, which are the building blocks of DNA. These base pairs are organized into about 20,000 to 25,000 genes, which code for the proteins that carry out the functions of the body. The human genome is distributed across 23 pairs of chromosomes, with each chromosome containing many genes. The Human Genome Project, HGP, was an international scientific research project that was launched in 1990 with the goal of sequencing and mapping the entire human genome, which is the complete set of genetic information contained within human cells. The project was a collaboration between scientists from around the world and was coordinated by the National Institutes of Health, NIH, and the Department of Energy, DOE, in the United States. The HGP involved the use of advanced technologies for DNA sequencing and analysis, and it took over a decade to complete. In 2003, the project was declared complete, with the entire human genome sequenced and assembled into a reference sequence. The reference sequence is a highly accurate and comprehensive map of the human genome that has become an important tool for researchers in many fields of science and medicine. The completion of the HGP has had many important implications for science and medicine. It has provided a wealth of information about the structure and function of the human genome, and has enabled researchers to identify many disease-causing genes and genetic variations. It has also led to the development of new diagnostic tests and therapies for a wide range of genetic diseases and disorders. In addition, the HGP has paved the way for new research in fields such as personalized medicine, where an individual's genetic information is used to tailor medical treatments to their specific needs. DNA can be damaged by a variety of environmental factors, including exposure to radiation, chemicals, and other agents. This damage can take many forms, including changes to the DNA sequence, breaks in the DNA strands, and alterations to the structure of the DNA molecule. Such damage can have serious consequences for living organisms, as it can interfere with the proper functioning of genes and lead to the development of diseases such as cancer. One of the primary sources of DNA damage is exposure to ionizing radiation, which can cause changes to the chemical bonds that hold the DNA molecule together. This damage can occur from exposure to sources such as X-rays, gamma rays, and cosmic rays. DNA can also be damaged by exposure to certain chemicals, including mutagens and carcinogens, which can alter the structure of the DNA molecule and cause mutations in the genetic code. Other environmental factors, such as certain viruses and even natural metabolic processes within the body, can also lead to DNA damage. To protect against DNA damage, living organisms have evolved a variety of mechanisms for repairing and maintaining the integrity of their genetic material. These mechanisms include DNA repair enzymes, which can recognize and repair damaged DNA, as well as checkpoint pathways that can halt the cell cycle to allow time for DNA repair before cell division occurs. Despite these protective mechanisms, however, DNA damage and mutations can still occur, leading to a variety of diseases and disorders. DNA can be extracted from a wide variety of biological materials, including blood, saliva, and hair. These materials contain cells that contain DNA, which can be extracted using a variety of methods. One common method for extracting DNA involves breaking open the cells to release the DNA, and then purifying the DNA from other cellular components using techniques such as centrifugation, filtration, or column chromatography. The resulting DNA can then be analyzed or used for a variety of purposes, including genetic testing, forensic analysis, and medical research. Blood is a particularly useful source of DNA, as it contains a high concentration of white blood cells, which are nucleated and contain DNA. Saliva is also a useful source of DNA, as it contains cells from the lining of the mouth that also contain DNA. Hair is another source of DNA, as the roots of the hair follicle contain cells that are nucleated and contain DNA. Other sources of DNA include tissues such as skin, organs, and bone marrow. The ability to extract DNA from biological materials has revolutionized many fields of science and medicine, as it allows researchers to study the genetic material of organisms and gain insights into the molecular basis of many diseases and disorders. It has also led to advances in forensic science, as DNA evidence can be used to identify individuals and link them to crimes or other events. DNA can be used to identify evolutionary relationships between different species, including both living and extinct organisms. By comparing the DNA sequences of different species, scientists can infer the evolutionary relationships between those species and reconstruct their evolutionary history. The basic principle behind this approach is that the more closely related two species are, the more similar their DNA sequences will be. By comparing the similarities and differences in DNA sequences among different species, scientists can construct phylogenetic trees, which show the evolutionary relationships between those species. This approach is known as molecular phylogenetics, and has become an important tool in fields such as evolutionary biology, ecology, and conservation biology. One of the advantages of using DNA to study evolutionary relationships is that DNA is relatively stable over long periods of time, meaning that it can be used to study the evolutionary history of both living and extinct organisms. In fact, DNA has been successfully extracted and sequenced from a wide variety of fossils, including those of extinct species such as Neanderthals and woolly mammoths. Overall, DNA provides a powerful tool for studying the evolutionary relationships between different species, and has led to many important discoveries in fields such as evolutionary biology, ecology, and conservation biology. The CRISPR-Cas9 system is a powerful tool for genome editing that allows for precise changes to be made to DNA sequences. CRISPR stands for unquat, clustered regularly interspaced short palindromic repeats, and quat, which refers to a specific type of DNA sequence that is found in many different organisms, including bacteria and archaea. Cas9, on the other hand, is a protein that acts like a pair of molecular scissors, cutting the DNA at a specific location. The CRISPR-Cas9 system works by using a small RNA molecule called a guide RNA, GRNA, to direct Cas9 to a specific location in the DNA sequence. Once Cas9 is bound to the DNA, it cuts the DNA, which can then be repaired by the cell's own repair mechanisms. By providing a modified or alternative DNA sequence during the repair process, the CRISPR-Cas9 system can be used to make precise changes to the DNA sequence. The CRISPR-Cas9 system has many potential applications in fields such as medicine, agriculture, and biotechnology. It can be used to correct genetic mutations that cause diseases, create genetically modified crops with desirable traits, and study the function of specific genes in living organisms. However, the use of the CRISPR-Cas9 system also raises ethical and safety concerns, and its use in humans is heavily regulated in many countries. DNA can be artificially synthesized in a laboratory using a technique called DNA synthesis or gene synthesis. This process involves chemically synthesizing short DNA fragments, which are then assembled into longer fragments using specialized enzymes called DNA ligases. DNA synthesis has many applications in fields such as genetic engineering, biotechnology, and synthetic biology. It can be used to create artificial genes or modify existing ones, which can then be inserted into cells or organisms to alter their function or properties. DNA synthesis can also be used to create large DNA constructs, such as whole genomes or chromosomes, which can be used to study gene function and regulation, or to create new organisms with novel properties. However, the use of artificially synthesized DNA also raises ethical and safety concerns, particularly when it comes to creating organisms with potentially harmful properties or unknown effects on the environment. As a result, the use of synthetic DNA is heavily regulated in many countries, and researchers are required to follow strict guidelines and safety protocols when working with synthetic DNA. The study of DNA has had a profound impact on many fields of science and technology, including medicine, agriculture, and forensics. In medicine, the study of DNA has led to many important discoveries, such as the identification of genes that cause or contribute to diseases, the development of new diagnostic tests and treatments, and the use of personalized medicine based on an individual's genetic profile. DNA sequencing and analysis have also helped researchers to understand the genetic basis of many diseases and disorders, and to develop new drugs and therapies that target specific genetic mutations or pathways. In agriculture, the study of DNA has led to the development of genetically modified crops that are resistant to pests or have increased yield, as well as the ability to identify and track the spread of plant diseases. DNA analysis has also helped to improve animal breeding programs and to identify genetic markers for traits such as disease resistance and meat quality. In forensics, the study of DNA has revolutionized criminal investigations by providing a powerful tool for identifying suspects and linking them to crime scenes. DNA evidence is now routinely collected and analyzed in criminal investigations, and has led to many successful prosecutions and exonerations. DNA analysis is also used in paternity testing and to identify human remains in cases of mass disasters or war crimes. Overall, the study of DNA has had a transformative impact on many aspects of our lives, and continues to drive new discoveries and innovations in science and technology. DNA is just one part of the complex molecular machinery that makes life possible. While DNA contains the genetic information that encodes an organism's traits and characteristics, it is only one component of the larger system that allows living organisms to function. Other important components of the molecular machinery of life include proteins, which are responsible for carrying out many of the functions encoded by the DNA, as well as various types of RNA molecules, which are involved in processes such as gene expression and protein synthesis. Additionally, the cellular membrane and other organelles within the cell play important roles in regulating the movement of materials into and out of the cell, as well as in maintaining the overall structure and function of the cell. Understanding the complex interactions between these different components of the molecular machinery of life is an ongoing area of research in many fields, including biochemistry, molecular biology, and systems biology. By studying the interactions between DNA, proteins, and other cellular components, researchers hope to gain a better understanding of how living organisms function, and to develop new treatments and technologies that can improve human health and well-being. Thank you for watching, subscribe our channel and see you in next episodes.